Thank you for coming. My name is Lew Lim from Vielight. And as in all scientific presentations, I should make some disclosures. That is obvious, right? I'm with a company that manufactures photobiomodulation devices, so there is conflict. But if you hear anything that I'm saying, you say there's a bias. Well, here's the reason. Okay, hopefully you just give me some slack and say this guy knows what he's talking about. We have some devices that are medical devices, but generally I say in this world of photobiomodulation, most devices are in a category of low risk general wellness. So they're kind of like consumer devices. FDA knows about this, and as long as we don't make a medical claim, so we kind of are careful about going there. Now, the takeaway really is, hopefully at the end of this, and if you have familiarity with photobiomodulation, you have some tools to ask questions, like somebody may say this wavelength is better than the others, and there's silence about power density and stuff. Then you have some tools to, in your mind to make some references and see whether you can discriminate between fact and non-fact. But when I present this, I hope I don't sound biased, because we really do a lot of work in coming up with the data, and we do a lot of measures, not just theories. Now, this is the many methods of delivering near-infrared light or red light or whatever to the brain. This is one method. I was going to show you, but my hands are full. So look at the pictures. We have a booth upstairs, and basically the LEDs underneath, and they point into, for us, we point specifically to some areas of the brain. And there are some theories about it. You get, helmets, like construction worker helmets that have LEDs underneath. You can have wraps. But we want to deliver enough power, and I'll show it to you later why power density is important and why certain things work better than others. But in principle, I say photobiomodulation does work, even little power, but we want to be better in getting outcomes from the brain. Now, another method is through the nose. When I first came out with this idea of stimulating the brain with the nose, and I found a patent for it too. Even people in my field thought this is crazy, because it's so novel, it is hard to fathom. But think about it. The olfactory bulb is part of the brain. It's just right above the nasal cavity. And I'll show you how effective the intranasal pathway is. Now, we do a lot of studies, and some of them are published. Most of them are in small case studies in clinical trials, case reports, a bunch of things, But I say that in this world of transcranial photobiomodulation stimulating the brain, there's a lot going on. There are familiar names of which universities are doing it. But I say we collaborate with a lot of research institutions and try to tease out what is the best way to stimulate the brain and draw significant response from the brain. So we cover, and I'm probably missing some of these, traumatic brain injury, where I would say personally, the strongest data is coming from. We are yet to do a big, just pivotal study with FDA, and so on, but that's coming. But even on double-blind studies and controlled reports, they are coming out really strongly. Dementia and Alzheimer's, We've been involved with this. If you follow us, you follow me, you know, we've been in this for quite a number of years. So there are some strong data. I don't have the time to show everything. But I'll give you the principles behind this in this presentation. There is a brain fog. We did a COVID study approved by Health Canada. It works for acute COVID. But unfortunately, acute COVID is not a thing anymore. So now it's long COVID. People are having this, problem with brain fog and stuff. So we are analyzing the data that's coming out of a pilot study in Florida. We've recruited everybody, but the assessment period is 30 days. So we're waiting for that for the last couple of patients, and then we do the analysis. Then we'll see what happens. Doing depression study, PTSD with the Boston folks, addiction at West Virginia, and actually even in Boston, Parkinson's, stroke. Now, the non-clinical side is also very interesting to us. I will show you some of the stuff that's coming out of our meditation study. There is tomorrow I'm going to talk about improving intelligence with photobiomodulation, and then reaction time in athletes, and also we're exploring consciousness, all this fancy stuff, but that is the versatility of photobiomodulation. Now, if you're not familiar with using light to stimulate the brain, it is really part of the electromagnetic spectrum, coming all the way from gamma rays, very, very, very high energy, very short wavelength. And as you know, gamma ray going to X-ray actually can modify the genes in the body. So it is high risk. But as you go along the wavelength, as they become longer, you go into ultraviolet light. And then in the areas that are in our territory, visible light, particularly on the longer part of it, the red, and then near infrared, this out of this picture here, then you know it covers radar, television signals, and so on. So imagine that the electromagnetic spectrum with very long wavelength can penetrate everything, right? But it doesn't mean that it can do anything for your brain, because it needs to be in a certain, what we call action spectrum. So for the brain, we found that near infrared light, mainly in the region of even visible red, but around 810 nanometers to 1100 or so. So that's where we are. If you go to photobiomodulation presentations, many people like to use this picture. It's colorful, it attracts attention, but it is not necessarily correct 100%. But it gives you an idea. Now follow the red. That's where it's supposed to activate the mitochondria in the electron transport chain to produce energy and so on. Now that is in the range of visible 600, which is kind of like orangey red, to 850, which is almost, actually it should be invisible, but you see that in the LEDs because LED is not like laser, it's got a bell curve. So it covers some visible spectrum as well. Then we go into 1064, 1070, longer. And if you look at the picture, it's on the left side, it shows a light beam activating anything but the mitochondria, which is not correct. Actually, just oversimplifying it. But that's a general idea. Different wavelengths activate different things. Now, some of you are maybe in this field and you hear this claim that 1070 is better than 810. 810 is better than 1070. When we talk about 810 or 1070, really it all came out from the earlier work using lasers. and you want to get like pure lasers going through, there are, in this spectrum, there are mainly two choices, 808 nanometer or 1064. So 808, the nearest, and we get all this great data coming out from the earlier research. So then the nearest LED we get is, okay, a nice rounding number is 810. And then the territory of 1064 laser, there is coming out from Legacy on cell culture study, 1070 or 1060. There's so much marketing going on with 1070, they don't say 1060 or 1040 anymore. But in reality, although it's like, they say it's 1070, most likely the LED they're getting is 1040 because it's more readily available, off the shelf, and you can get more power of 10 from 1040 to 1070. That's the trade secret. Now, what do the computer modeling say? It is popular actually among academics. You do like a PhD or a PhD student or a postdoc, you really love computer modeling. It's really, but and then it gives you like a simulated outcome saying how the brain is responding to the light stimulation. But really, what is it saying? Historically, computer modeling, mainly Monte Carlo simulation, to use that, depends on the variables you input into the model. The brain is really, really complicated. And we have a paper that is pending publication or under review right now using computer modeling together with University of Toronto and Baycrest Hospital. It is showing certain data. It's a really great exercise, but I hold my, okay, based on this data, it is what it's saying. But I say historically, you can, mainly the weight is on using 810. 810, according to the modeling, penetrate deeper. And I've depended also somewhat in the early days on modeling. And there's only one paper that says 1070 or 1064 might be better. So what do lab experiments tell us? Now, this is another set of data that is produced by the lab of Juanita Anders. She's been in the field for a very long time. I think this was data that came out in 2013, 2014. And but she never published it. She did this work with some investigators in the FDA who are also in this kind of research, do animal studies. And God knows, Dr. Anders has sacrificed so many animals to arrive at this, but she did this using covert. And her team directed light through the covert with different wavelengths and see what penetrates deepest. Now you got to remember, there is absorption by water. The longer wavelength get absorbed by water, and I've said before they could be outside of the action spectrum, of photobiomodulation. And her conclusion is, say, Lew, I talked to her and said, okay, Juanita, what would you do? I'm talking about like 10 years ago. She said, just go for 810 nanometers. And that was the old days. But if you look at it, you can see that 810 has got the highest penetration to use arbitrary units. And even 1100 or 10, if you like 1070, shows pretty good peak there. But her conclusion is 810. Now, what about, there is this theory. You want to activate the electron transport chain, the cytochrome C oxidase I mentioned before. And from their work, 810 seems to be the one that's activating the cytochrome C oxidase the most. Now, what is cytochrome C oxidase? It is you can call it a photoreceptor, an enzyme in the last terminal of the electron transport chain. You can activate it through red and near-infrared light, and then it gets produce more ATP, gene transcription, and a bunch of things and release nitric oxide back to the body. So there are good things coming out of that. That is the general principle. I don't want to go too deep into that because of other things I need to talk about. So really, you want to come down to what is the difference between 810, 1070 when you hear this? We are seeing data that's beginning to tell us the difference. I'll go into that a bit later as well. So 810, I would say generally activate mitochondrial activity. 1070 does that, but it is activating a physiological response because of it's going through the light-gated ion that is receptive to light and heat, which is TRPV1, calcium ions, which is a complicated process, and also the heat shock protein as well, I mentioned here. So there is some difference. Now, what do? I think, although we use a lot of 810, in the future, I haven't actually decided whether we want to go and use this longer wavelength or 1064 because we're still doing the research. And I'll show you. And I kind of feel that we need to use both. And they are both useful for conditions like traumatic brain injury and for Alzheimer's disease based on animal studies. So I'm exercising a bit of patience here, but in a few months, I think we will release and I'll make some statements about that. Now, what are the outcomes? The beauty about photobiomodulation, the questions sometimes get asked is, how is it possible that you do this and you make so many claims? In truth, we try to avoid too many claims, but the fact is, when you are able to modify something fundamental, like what's happening in the mitochondria, the electron transport chain, it just kind of goes up to a higher level of tissue modification and so on. It's on mainly in the modification of the transcription of the genes, if you like. So many areas, it covers, and these are all relevant if you study the pathophysiology of traumatic brain injury, Alzheimer's, whatever. It actually involves multifaceted, multiple aspects of pathophysiology, from synaptogenesis to creation of new synapses, angiogenesis, which is really your blood vessels, creating new blood vessels, increasing blood flow. The big thing also is anti-inflammation, anti-apoptosis. Apoptosis is the programmed cell death, is kind of controlling that. Increased neuron progenitor cells, the stem cells, in creating new neurons. Increased SOD, superoxide dismutase, which is to neutralize free radicals. And then you got the BDNF, GDNF, they're really growth factors in the brain. And very important but forgotten is actually to control the excitotoxicity. Every time your brain processes something, a byproduct is some kind of toxins and it's got to be cleared. So I'll summarize. I'll say the state of knowledge today is mainly, put it very simply, 600 to 700. You see this in the body panels. They do this visible rate. You see bright red is usually 600 something nanometers. Then interesting thing is nothing happens between 700 to 760. This is very interesting. It's like a blind spot. So suppliers of photobiomodulation devices should not be saying, I've got something at 750. No. I think there's enough data there for some reason. And then 760, 850, I mentioned mainly in the cytochrome C oxidase, the enzyme in your electron transport chain. Then 800, 850, 1500, you still work on cytochrome C oxidase, but more in the water structure, the TRP1 receptors and calcium ions. Then there is a small group of people in this field say that, no, you got it all wrong. It's not really what's working on a cytochrome C oxidase. I did my experiments. It doesn't seem to be that. So he's going against the tide. What he's saying is that photobiomodulation decreases the viscosity of water. He did this nano material experiment. He's got something there when he shone laser in the red spectrum. It sinks in because the water is less viscous. So what happens is your ATP synthase is able to function better because there's more, it's less dense or not so viscous. Now, top of parameters, I don't want to make this look really complicated. But the main thing here is when you talk about parameters, it's really coming up with the dose. How much dose you're going to put in and what kind of, and the dose is a function of power and time. So these are really the two main elements, when it comes to dose. And the field, because of history, because they see this optimum dose of 3 joules in the cells. So we try to kind of do our other parts of it, power and time, and fit in. I think that's wrong. And I'll show you why. You really ought to start with how much power you need, power density in the area for penetration and for activation, and how much time. I'll come to that. So power should be related to density, because these lights are really powerful, but they're not reaching you because it's all spread out, right? I'll cover that later. So you want penetration, calculate by how much concentration it is. Time here means duration of time for biological action, penetration, how much dose, I mentioned that before. The beam spot size, the concentrated area is important. The pulse frequency is an area that I'm passionate about because of these new data is coming out. And the other parameters like LED positioning, which I think is important. And let's start with dosage. This is really a puzzle. From a little cell of 3 joules, then people are trying to figure out, okay, how much dose do I need to do to get is optimum? Here's one study that made a claim, they did this protocol, and then 3 years later, they published to see mainly looking at these others metrics, which are, how much pain you're experiencing during your training and exercise, muscle contraction, and soreness. And they found that you can't test so many, but with what they tested, they found that 100 milliwatts was ideal. And do it for 100 seconds, so you get a figure of 10 joules. But they didn't pause, it just was continuous. I just want to show this as an example. Then one thing is also important is you will find that if you're treating your patients or doing these experiments, you get more response from those who are impaired in some way, as opposed to those who are completely healthy and you say, oh, this thing doesn't work, I don't feel any difference. That's because you're like, you're all jacked up and you're functioning in optimum. So there is a homeostasis level for everything. That's how your body functions. So the more you're outside of homeostasis, because of maybe like oxidative stress, the more you respond. And one thing about photobiomodulation is it creates free radicals, reactive oxygen species, ROS, that kind of trigger a response from your body, but at low level, right? So when it's too much of it, it overwhelms the body. And I'll come to this quite an important principle in photobiomodulation. What is the ideal? There's a window. Too little doesn't do anything. Too much, as I've shown you, create this body of free radicals that your body takes time to clear. So there's a window somewhere. Now, what is it? That's where we have to do our clinical studies. Now I'm going to go through some discoveries that we've come across. We've been doing these are actually all ongoing. Use real-time fMRI to see what is really happening to the brain when you are directing laser to the brain. We are experimenting with living cells and cultured tubulins and microtubules. Microtubules are actually polymerized from tubulins, so they're all pretty important. There's quite a lot of discussion in the world of consciousness, and particularly with Stuart Amarov and that particular group, this quantum physics in the consciousness, and the secret lies in the tubulin. I don't know. I'm looking at quantum physics, and I think there's a lot of hypothesis and speculation, but there is something there. Now, mainly because our brain is kind of warm and wet, it is not conducive for quantum physics. But anyway, that's an aside. And then we're doing meditation. Now, this is the thing, we've got some really interesting, like supernatural outcomes from our work in studying meditation. I'll come to that. That makes me think, are they right? Is it, quantum physics? We'll talk about it. So I'm coming to another element, power. Say power density matters. And this is this Siemens MRI scanner that we used in Baycrest Hospital. As an engineer, we put a laser. It's got to be laser through a glass fiber so that it's compatible. So one here first and then one through the nose. Let's see what happens. Now I got to say that this is not published yet. There is a poster presentation that was done in a imaging conference. We are recruiting like 45 subjects. We are, I think, about halfway through right now. It requires a lot of analysis, but I've got preliminary data from a couple of subjects, and this is what it's showing. For this person, the optimum power density is 150 milliwatts. If you want to produce the most response from the brain, and the response from the brain is showing in, you can say there is differentiation between cerebral blood flow and BOLD, which is blood oxygen level dependence data. The idea is 150. That's a lot. That is a lot of power, and no off-the-shelf LED can give you that kind of power. That's why we do the LEDs like we do, because we want to be able to deliver that kind of power. But this was really surprising to me, because there's so much coming out in various studies. 20 milliwatts is enough, and so on. So this is a very interesting lesson. I don't know when we finish with the 45 subjects, what it's going to look like. I suspect it might be lower than this. Now this is interesting too. This was done with, I think, the 1064, wavelength. And we pulse at 10 hertz and 40 hertz, alpha and gamma. And look at the peak. Look at the 40 hertz on the left half and the right side. The peak is 150 milliwatts from there. But as you increase the power to 250 milliwatts, it's gone negative. You get a negative response by having too much power. But if you don't have enough, there's nothing in the brain. Okay, I have to qualify and say this is looking at the blood perfusion, the cerebral blood flow, BOLD imaging in the brain. At 10 hertz is different. When you pulse at 10 hertz, it's different from 40 hertz. That's why I say pulsing frequency matters, because if you're going to be more precise, you got to look into this. And where is our peak? At 10 hertz, the peak is 175, even higher power. Okay, so the summary here is power density matters. And there's a biphasic response saying that too much is no good, too little is not enough. Then this is another way of looking at it. And just differentiating between 150 milliwatts and 175, 150, that difference in 25 milliwatts seems to matter.
This is blood flow? Is that...
There's both blood oxygen level dependent imaging. It's quite close to blood flow. So power density matters. Now, then we added the intranasal to the left side. So we're not just saying, you know, if you look at the previous imaging, you can see there's more on the right than the left because we have one here. And now we do the intranasal, the whole brain is kind of evenly responsive, it's more global. So the intranasal does make a difference. And this is just the intranasal at 810 nanometers. So we pulse at 10 hertz and 40 hertz. And at 10 hertz, you see that 9 milliwatts. Okay, there, transcranial, we talk about 150 and so on. This is just 9. Look at the amplitude, what is it? The Bohr percentage is 2.5 and so on. Okay, with 9 milliwatts. And I go back to transcranial and look at the peak 2. So get a systemic response, okay? So the intranasal actually is pretty powerful and is logical because your brain, that is presented, the olfactory bulb is part of the brain. It's just so close, you know, and you don't have to go to the scalp, the hair, and everything, right? So this is like minute by minute, and we see really, great response from the brain. I've been looking at the literature for other form of brain stimulation, transcranial magnetic stimulation, and some other electrical stimulation like DDS, DDCS, TACS. Actually, there's nothing like this. While you don't feel any effect, the brain is really responding. So in summary, looking at what's now, 6 months later when the data is up, I gotta say this might change. Okay, I just have to say that, but this is what we see right now from the work we're doing. Now we want to see power between like 75 to 175 milliwatts generally because, to allow for some differences in people. Intranasal is only like 7 to 10 milliwatts. Okay, we use a laser there. The wavelengths 808 is mainly physiological. 1064 would be drawing, like more presentation in the blood flow. And pulse frequency matters. Now, so I research. So I also review articles and I write papers. And I say some of the papers that are published are actually flawed. And that's because there's not enough knowledge in the industry. And here's one, okay? If the audience are watching, I'm sorry, I gotta bring this up. So they form a conclusion like near infrared of 15 to 30 milliwatts brought more treatment improvement than 40 to 90. I tell you what, because most of the LEDs out there are mainly between 15 to 30, not too many between 40 to 90. And it's very rare that ours, that we are trying to develop very high power so we have the room to do it. We can go up to 300. Okay, and then we kind of pare it down and come up with the right set of parameters. So this literature, they're flawed in analysis or they're outdated because when we start to release this data, it's going to look different. And a final detail is important, the contact. It's just logical, right? The further away you are from a source of light that is kind of divergent and diffuse, like light bulb and everything. It gets dimmer, less energy reaches your skin. So, and the formula in physics is really, you literally square, you have this ratio, you square as a denominator by the distance, so it's exponential. And we've been measuring LEDs that came off the shelf, like 25 milliwatts, and see what some devices are using. By the time it reaches your scalp, it's just 5 milliwatts. So your question then is, does it work? I don't know. It may not penetrate that much, but photobiomodulation does draw a physiological response. But there's a Parkinson's study going on. They came up with analysis that there's so much placebo effect. Actually, any medical device, this is a problem, that medical devices have when you try to get FDA approval. The placebo effect is really, really strong. You do something, you do some work, put this stuff on your head or whatever, a lot of it comes from placebo. So we try to tease out what is placebo and what is real. And sometimes a lot of the studies are self-reporting. So I'm feeling this, I'm feeling that, you do your depression scale and your cognitive scale, you're self-reporting. But I think imaging is where the difference is going to be. So just be conscious of that. Now okay, if you want to overcome distance, then you use lasers. Lasers collimated. That's why they tell you, do not shine the light on a plane. They're like miles up there, and they still get, they get affected because lasers can travel a long way without attenuating. But LED is like small distance, it just loses its power. So this is just a way of showing it that the formula is there, and the attenuation by distance. So contact, if you're using LED, try to get contact as much as possible so that the speck you're seeing is really what you're getting on your tissue. Measure. We do measuring all the time. Now, time duration is part of the equation. Now, those, like I say, depends on the power and the amount of time. How much time do you need? Too much dose, I say, is too much. You get these free radicals all over the place. And too little, nothing much happens. This is an analogy. That's how I cook my turkey. When you're like impatient, you put blast with a lot of power in 5 minutes. Okay, it's not cooked inside. So you do like, okay, give it time at a lower power and get it evenly cooked, right? This is actually a very easy way to think about it, but there is a more scientific way to think about it. Now, this is going to be complicated. It involves DNA duplication. The double helix was discovered, I think, in 1956. So a couple of years later, these folks at MIT try to explore how much time do we. So if you want to see physiological response, it's really actually you're talking about gene transcription, DNA replication, and so on. So this is really on a sample of E. coli bacteria, and very simply, they came up with a nitrogen isotope that's heavier than the normal, which is N15 versus N14. Put into the environment, as the cell keeps going to the next generation, they get to be with the environment, which is N14 light isotope. Now, this is the process. Each generation where they see changes is 20 minutes. So to them, based on E. coli samples, it takes 20 minutes to do DNA replication to get, for us, I'll see as, physiological effect. But in reality, in a human being, it takes hours. So that's why regular use does make a difference. So this is a scientific reason. Somebody asks you, actually, I'm presenting this for the first time. I'm not touched on this before because I'm given 90 minutes, so I have more time. So this is how, that I think that amount of time is kind of good to use. Now, pulsing is really important. And now when we did, we started to look at tubulins, microtubules. They're like, okay, we are unlocking the door into something we don't know, and say, okay, intuitively, I think if you look at what's happening to tubulins and microtubules, because microtubules support your structures, right, exons and all that. So it's worth looking at. Then we did, and also living cells. The living cells, like basically healer cells, cancer cells, that's cervical cancer cells and glioblastoma cells, and see what the effects are. But to introduce microtubules, basically it is important in all cells, not just exons. They support the structure of the cells, and they're involved in signaling too. That's what tubulin looks like, and you can get all detail with it too. But here is what we find. We actually published this, and this was using 10 hertz. Now, the interesting thing is, on the right side, you see the control of these tubulins in the solution is kind of, they're present when we stimulate with 10 hertz. Actually, they kind of disappeared. So what 10 hertz did was it caused depolymerization. It's like opposite effect. Isn't that interesting? So when we did the versus control, there is the looking at the optical density. It's actually less. You can see from the microscopy. Now that is really interesting. Just hold your thought there. But on the other hand, there's some dichotomy here that, when we did on the living cells, there is a more resistance. These are cancer cells. We directed electrical. They actually do that in Japan. To treat glioblastoma, some physicians, I don't know, they do that into glioblastoma and try to diffuse this the cancer cells. But we see resistance when we shown 10 hertz photobiomodulation at 810 nanometers. They show resistance too. While in the meantime, they allow more currents to flow through. I don't understand it, but the evidence is there. So it improves the health, and yet, it does this thing. It depolymerizes. Now we got some initial data. What about other frequencies? Okay, we are still doing this. Now, when we did 40 hertz, it's different from 10. Actually, that's 40 hertz and then 1000 hertz. It starts to shift the other way. So it seems to polymerize at a higher frequency as well. So 40 hertz seems to be like a switch, the gamma. I say that because there's some other work done where this lab in University of Alberta under Jack Tudinsky, they did this work and found that 39 hertz, actually not 40, allow more electrical signaling through 2 points, the neurons. There's a lot to be discovered, but this is something that's kind of revealing. We're doing this work still. Then this is earlier work. They found that at a higher power, this photobiomodulation look at what's happened to for pain signaling. Actually, higher power blocks. Higher power actually blocks pain signaling, which means that it is inhibitory. So try to relate back to the imaging I showed. Higher power, get negative effect. So it's inhibitory, but it's good for blocking pain. Okay. Now this paper is under review right now using Raman spectroscopy to see what the changes are in the protein structure. Just to summarize, your microtubules have the secondary structures, involving the the alpha helices and beta sheets. And we found that the 10 hertz actually is causing a transition. And then we said, okay, maybe this is relating to conditions like trying to treat Alzheimer's disease, where you want to clear the beta amyloid or deal with the neurofibrillary tangles. This is good transition, you know. Then you do another intervention, and I'm hypothesizing here. I don't know what 6 months is going to be from now is going to look like, but the inhibition may actually consolidate the improvement in the tissue structure or the microtubules. So this is an important discovery that that photobiomodulation at 10 hertz can cause a transition in the microtubules is open to new treatments. Now this is the summary. I think we can skip this. Now I talk about more specific pulse frequency like gamma. This is EEG, if you guys are familiar with this, and there are two groups. One is eyes open, eyes closed, they're pretty similar. But the characteristics are actually different between young adults, healthy aging adults, and those with Alzheimer's disease. The gamma is really low. As you age, the gamma is shifting from the normal 40 hertz or around that to a lower frequency. So it is a case for us to try to restore gamma so that perhaps the brain functions better. Now, gamma is also found to be important in memory encoding. This is the research done at UCSF. They look at subjects with seizure risk. So the hypersynchrony in the network. Now, it found that when gamma is present, the spikes and you can translate that to the risk of seizure to be lower. And in the meantime, the memory encoding is higher. So gamma has a dual effect here in reducing seizure risk and helping with memory encoding. Now, this is a famous landmark study published at MIT, looking at pulsing 40 hertz into your eyes and see what happens in the animals. The transgenic mice that are bred with the markers for Alzheimer's disease actually had their beta amyloid load reduced after 7 days. And then the microglia were modified from inflammatory to non-inflammatory. So the non-inflammatory were kind of maybe helping to clear the amyloid beta. Then a couple of years later, they directed sound at 40 hertz too, and found that actually more of the brain respond. A few months ago, they said, actually earlier we didn't understand how this works. A few months ago, they published another paper in Nature too, and said, 40 hertz actually seems to help the lymphatic system to clear the deposit involving the aquaporin, the astrocyte channels. So that is an explanation. That is great for us. So we want to try to get gamma into the brain. So MIT is MIT. They're really strong and powerful. Everything they say, a lot of people follow. But I thought this is a really interesting study. Came out of China. That, look, we did a study at 10 hertz on the mice. They are, again, we get better behavior, and they compared, it's too small, but I'll say that they compared 10 hertz and 40 hertz. That is more effective in clearing the beta amyloid. Then there's another group in New York under another famous neuroscience lab, Dr. Berserk, anyway, that did and found that 40 hertz they replicated the study from MIT. It said it doesn't show anything. So there is a debate. I have to say, there's a debate going on right now in the field of neuroscience, how well 40 hertz work well, it does not work, and it doesn't work, and so on. But it is, but I say 10 hertz from this study shows that it works too. And I can say that from our earlier study that we published, our first Alzheimer's or dementia study that we published, we used 10 hertz. It seems to work. Then UCSF did 40 hertz, it works, along with fMRI. So they both work. And so pulsing is revealing its relevance for treating neurodegeneration. So we can't say pulsing doesn't work. It does work. Now we got to refine how. Now, does it train? So I talked about, okay, we want to get gamma in the brain. Can we do it? So this is an ongoing study now, okay? I will differentiate this and from the meditators, but it's consistent. We published a paper in 2017 showing that when we directed gamma into the brain at 40 hertz, alpha, beta, gamma amplitude increased, delta theta reduced. Very, very interesting. It was a very interesting discovery, well cited. Now we're doing this. Okay, let's get more subjects and just do gamma on healthy brains. And it's pretty consistent. So this guy, the first line is the baseline, and then pre-treatment, this is the treatment, and this is after treatment when you stop for, I believe, 5 minutes, and then post, was it 15 minutes later. So these are the net effect. So this minus baseline, you get, just before the treatment, he had higher low frequencies and and the and low gamma. But after real time, these are real time data. We actually measure the EEG while we're doing the intervention. His gamma has gone up. While 5 minutes is very interesting. After we stop, the other waveforms went down, but the gamma maintained. And then 15 minutes later, after we stop everything, the brain restored and the gamma keeps going up. This is pretty much, everybody vary a little bit, but generally at the moment, what we're seeing that when we deliver, if you want gamma in the brain, deliver gamma through photobiomodulation at 40 hertz, and this is what you get. So I'm waiting to finish all this analysis and then publish. But this is just 5 minutes, I talk about 20 minutes. So why do we need 20 minutes? Actually, go back to cooking the turkey and the gene transcription for physiological changes. This is where you differentiate between reading electrical signals or blood oxygen level dependent imaging or cerebral blood flow. This is nice to see that the brain's really responding. But I think to get physiological response, healing, recovery, I think you need a longer time. 20 minutes each time is kind of like, no, it can't be for specific, but the indications are there. Right. So this is another flawed study. Says there's no difference between pulsed and continuous wave modes. So this was a study, I think, on traumatic brain injury too. Yeah, it is. And I looked at they did a systematic review. So they did not include some studies that actually declare the pulses are very important. So sometimes it depends on when papers are published, you got to look at the methodology. But unfortunately, people don't have time to read. So you can form the wrong conclusion, Let me say categorically, pulsing matters. So I published this in February, a couple of months back. It's on the really a pathophysiology of what's happening in traumatic brain injury. I'll summarize and say it is multifaceted. It's not just the primary pathophysiology actually is, I'll say, the axonal. What's happening to the axon? That disconnected, disrupted. But there are a lot of other things involved, like the inflammation, and what's happening to synapses and to the neurons and so on. So go there. But the point I'm making is, and I go back to what I said, that photobiomodulation, although there's just one delivery, is multifaceted. It deals with all these facets of pathophysiology, which makes it really interesting. So go there. Now meditation, our is very our pet project at the moment. We have 2 meditation studies going on. One in Tucson, Arizona, they just finished, and they're looking at bliss states, different pulse frequencies from. Okay, we do a sweep, different pulse frequencies. We have the NeuroPro, which is a research device that does that. The meditators, these are long-term meditators. I'm not one of them, but these are like meditating for many years. They have a trigger at a high frequency, and they get into this bliss state or altered state. So that is telling that there's some resonance, and I'll explain a little bit about the mechanisms. And in Toronto, we're doing also on meditators, and I'll show you the data because they're kind of preliminary data available. So there is we did like treatment for 20 minutes, but you can just see the protocol here. And we look at the EEG, we have mystical questions, but we are also looking at the different pulse frequencies of 4 hertz, 10 hertz, 40 hertz, 120 hertz, and a sham. It's all over the map, right? This is actually, for us, it's kind of disturbing and also very interesting. It is showing that all these subjects are different. They respond differently. They're significant. They're all responding significantly away from a sham, but they're different. I'll show you. We look at microstates, and what we're seeing is you can't see it, but blue is pre, red is during, and the yellow is post. And post, actually, after we stop treatment, we see significant buildup in the response in the brain. But let's be more specific. Okay, this can be complicated. There is just taking 2 subjects. Okay, blue is delivering at 4 hertz. Red is delivering at 10 hertz. Yellow is 40 hertz. This purple is 120 hertz. The green is a sham. The different columns, we looked at the brain waveform delta, theta, alpha, beta, gamma. Very different, right? Let's take for example, this guy in gamma is off the chart, but we were delivering 4 hertz to get his gamma off the chart. When we deliver 40 hertz, pretty much very little is happening. Now this other guy is, you see his purple is, is of the chart. You look at his beta gamma states, so we're delivering. So he was responding to the purple, which was 120 hertz. They're different. And when you put them all together, like, okay, sometimes you cannot, total up the numbers, and all we know from total numbers is they're significant, but they respond differently. If you remember, I showed you the EEG on normal people, healthy brains. We deliver 40 hertz, you get gamma 40 hertz, the native gamma at a higher amplitude. But the long-term meditators are different. So there's much to say about people with supernatural brains. I don't think I have. I'm one of those. But and so we did a dipole analysis. It's preliminary on limited data, and we saw the clusters in one region, the posterior cingulate cortex. The posterior cingulate cortex is identified. Those who are familiar with Alzheimer's disease, the pathophysiology, is one of the main. It's said to be where Alzheimer's disease originates from. Some say it's posterior cingulate cortex, some say it's the entorhinal cortex. But anyway, the question is, there's cluster, and it looks like inhibiting. This is an interesting study on meditation. Posterior cingulate cortex is implicated in meditation, but success in meditation is inhibiting in that particular region of the brain. So it kind of tie in with what we're doing. And so it's telling us that 40 hertz actually is inhibiting. 10 hertz is kind of stimulating. Now, we have positioned the LEDs on the hubs or the nodes of the default mode network. Okay, there's like the dorsal prefrontal, precuneus, the posterior cingulate cortex, the PCC. From the nose goes to the entorhinal cortex. Now, something interesting happened a few years ago when we were starting to do these experiments on long meditators, and we decided to shift. I said, no, literature is telling us that we should not be stimulating the default mode network because meditators get into that state outside the default mode network, and we're pointing into the default mode network. So, okay, let's do this outside the default mode network. And we started shifting the modules. We did not get the same kind of outcomes. It was really, really puzzling. The only conclusion is that, but we were pausing at high frequency. Excuse me. It tells me that I've got 20 minutes left. So, yeah, and that is getting outside, so you want to be outside of the default mode network in meditation, but we've seen that outcomes. And as I was saying, okay, this is not working, it was very, very, very puzzling. So it can only be that when these meditators were responding to high frequencies, at that time we were already noticing they were responding at 40 hertz and above, like 120 hertz, some at 400. And our main investigator, he's a really very high level meditator. He likes 1000 hertz. That's very, very, very rare. And it seems to point, and it ties up with the other work we're doing. It seems that a high frequency is inhibitory, starting from 40 hertz. Gamma is like a switch. So we got out of the default mode network, which is what you want to do for meditators, and start moving into the salience network, where involve the anterior cingulate cortex, the insula, which are relevant for meditators. So that is an explanation, to the mechanisms of what seems to be happening to the brain. Okay, there's more work to be done. This is kind of tying in what's going on with our experiments. And this is a way of explaining it. So get out of it, get meditation blessed. Now, summary, just very simply, these parameters matter. I've shown a lot of details: power density, pulse frequencies, the time duration, and the intranasal. Here's a more summary here. So we're looking at power density for the moment from what we are looking at in the data, 75 to 175 milliwatts. per square centimeter. The intranasal is just 7 to 10. The duration is, for physiological changes, 20 to 30 minutes. And we're pulsing it, evenly at 50% duty cycle. And the alpha seems to be, remember I said transitioning the microtubules, so it's causing some kind of transition. It allows you to prime. It's like priming the brain. So EEG, I remember some of the neurofeedback practitioners telling me that, talking about neurofeedback seems to work better when we prime the brain with photobiomodulation at 10 hertz. I don't know what your experiences are, but maybe this is like, helping to get better results, better outcome from neurofeedback. And for high functioning brains, I would say get into the gamma state. I will touch on this more tomorrow. I have another session. And the default mode network stimulated at 10 hertz. And switching over to be inhibitory at around 40 hertz. Now there is opportunity here. Now we got a lot of data. We want to personalize it. So what we're doing now is to go into artificial intelligence and see if we can accommodate different brains, different times, but the input variable is going to be with EEG. So that's the stuff we're doing now. So thank you. Thank you very much.